Hello students, this is Aisha, a third year MBBS student. Today we are going to discuss about body fluids and circulation. It is another important unit in human physiology. Okay, so let us discuss in detail. First we will start with the introduction, that is oxygen, nutrients and other essential substances will be provided to cells. These cells will utilize the oxygen, nutrients and they will produce harmful substances. These harmful substances need to be eliminated, otherwise it will cause damage to the body, right? How these processes occurring, how those harmful substances are eliminated? It is eliminated through a fluid, okay, that is called blood. Blood is eliminating this. See, this oxygen will be there, no, through artery. This will be supplied to the cells. These cells will utilize the O2 and will produce the CO2. This CO2 will be carried away with the vein. Okay, all these blood vessels are there, no, it contain a fluid that is nothing but blood. This blood is the commonly used body fluid in the higher organism through which circulation will occur, and all the essential oxygen, nutrients will be supplied to the cell and harmful substances will be eliminated. Okay, not only blood is commonly used, but lymph is also helping in transport of certain substances. Okay, blood is commonly used, but lymph will also help in transport of certain substances. So this is the introduction. Let us know the overview of this chapter, body fluids and circulation. Here it is divided into three parts. I just made a flow chart for better understanding of the whole unit. So here you will first learn about the blood. Blood is the main body fluid, no, through which all the substances are transporting, no. So first we will discuss about the blood, its composition, like what all the elements are present, cells are present, and we will learn about the blood groups and RH factor, and we will learn about the blood coagulation method, how blood clot will occur when there is some injury on the hand or anything. Blood will flow, right? To stop the blood, how the blood coagulation or clotting method will occur, we will learn about this. And next we will learn the important part, that is circulatory system. In circulatory system we will learn about circulatory pathway, and about heart we will learn its structure, cardiac cycle and regulation and ECG, and at last we will learn about disorders. Okay, so first we will start about this, that is blood composition, blood groups, RH factor or RH grouping, and blood coagulation. Okay, first we will learn about the blood and its composition. See, blood is a fluid. We already discussed it is a fluid, right? Blood is a special connective tissue. You have to remember this. A question can come. Blood is a special connective tissue. What are the contents of blood? First of all, blood have a fluid matrix, that is plasma. Now blood will be there, no, it will be having a matrix, fluid matrix, that is called plasma. It constitutes 55% of the blood, and in the blood plasma we will find formed elements. Okay, formed elements will be there, they constitute 45%. Okay, remember the percentage. And next one is, let us discuss in detail about plasma, how plasma will look. See, straw color means light yellow color. Plasma will be light yellow color. And viscous fluid means not very, it has some consistency. Okay, like honey will be little bit, it's not so thick also, not so fluidity also, no. Honey is like little bit viscous. In the same way, this plasma in the blood will also be viscous and it is straw colored. Okay, now plasma constitute 55% of the blood, and in plasma also some contents are there. Let us discuss those contents. First one is water. Water constitute 90 to 92%, whereas protein constitute 6 to 8%. Okay. See, what are the proteins in plasma? First protein is fibrinogen. We all know that fibrinogen helps in coagulation, clotting of blood. Okay. Second one is globulin. Globulin means what? It is helpful in defense mechanism. If any external foreign body is entering the body, then this globulin globulin protein will fight against that foreign particle and hence helps in defense mechanism. Next protein is albumin. Albumin helps in osmotic balance. Remember this is so important. The proteins and its functions are very important. Fibrinogen helps in clotting, globulin helps in defense mechanism, and albumin osmotic balance. Okay, next, water constitute 90 to 92%. It also contain, plasma also contain some minerals, okay, like sodium, chloride, bicarbonate, magnesium and calcium, and some simple absorbable forms will be there, that is glucose, amino acids and lipids. Okay, this is the content of plasma. See, factors for clotting are also present in plasma. Yeah, plasma contain fibrinogen that helps in clotting, right? Factors for clotting are present in plasma. And one more thing you have to remember, see, in plasma, if we minus the clotting factors, what do we get? That is serum. Okay, plasma minus clotting factors, if we remove this, then the remaining will be known as serum. So this is about plasma. Now let us discuss about the other 45% of the blood, that is formed elements. Okay, formed elements are constitute 45% of the blood. What does it contain? Erythrocyte. Erythrocyte means red blood cells, and leukocytes. Leukocytes means WBC, white blood cell, and thrombocyte. Thrombocyte means platelets. Okay, you have to remember this its value. Okay, erythrocytes are 5 to 5.5 million per mm cube. Okay, it is the highest value. They are present abundantly. Erythrocytes, you can see by the value only, it is present in millions, 5 to 5.5 million, and leukocytes are least, you see, 6,000 to 8,000 per mm cube of blood. Okay, and thrombocytes average, not so very high, not so very less, that is 1.5 lakh to 3.5 lakh per mm cube. Okay, now let us discuss in detail about the formed elements. First we will discuss about the erythrocyte, that is RBC, red blood cell. See, red blood cells are most abundant among all. We already discussed it is most abundant. And where RBC is formed? RBC is formed in red bone marrow in adult. In bones there will be red bone marrow. In this red bone marrow, this RBC production will occur. Okay, see, RBC is enucleated. It has no nucleus. It has no nucleus, and it is biconcave in shape. See, from here also it is concave shape, and it is also like cave shape, concave, two sides, biconcave shape. And see, it has hemoglobin. Okay, that is red colored hemoglobin. I told you the structure. It has 4 heme part and 4 globulin part. Okay, globulin chains, and in the heme we find Fe2 plus iron. Okay, so they are red colored iron containing complex protein. This is complex protein formed element, that is hemoglobin. Okay, what is the, in healthy individual, 12 to 16 gram of hemoglobin should be there. in every 100 ml of blood. Listen, this is so important, this value. What in a healthy individual, how much hemoglobin content should be there? 12 to 16 grams of hemoglobin in every 100 ml of blood. Otherwise we will say that if the hemoglobin content is less, then anemic patient, no? So average 12 to 16 gram of hemoglobin should be present in 100 ml of blood. And RBC, it plays a significant role in transport of gases. Yesterday we discussed in respiration how RBC will play a role in transport of gases. What is the lifespan, for how long this erythrocyte can live? That is 120 days. After 120 days, it will die and new cells will be produced from bone marrow of red bone marrow. So where it will die? After 120 days it will die and they are destroyed in spleen. Spleen is an organ in which RBCs will be destroyed. That's why it is called graveyard of RBC. Okay. So we are done with erythrocyte. Now let us discuss about leukocyte. Leukocyte means white blood cell. White blood cell means white, means colorless. It doesn't have hemoglobin, that's why it lack red color. And unlike RBC is enucleated, no nucleus is present, but in WBC nucleus is present and it is short lived. Like 120 days it's not living, it is short lived, within few hours and days it will die. Okay, now let us discuss the classification. WBC has more number of different type of cells. Okay, let us discuss in detail, and it is also very important. Okay, so WBC is divided into granulocyte and agranulocyte. Granulocyte means what? The cell which has granules in them. Okay, agranulocyte means no granules. Okay, granulocytes are of three types, that is neutrophils, eosinophils and basophils. Neutrophils, why the names are like this? Eosinophils means it stains the acidic dye. It stains the acidic dye, that's why eosinophils. Basophils means it stains the basic dye. Okay, that is why basophil. Neutrophils means it doesn't stain either that and doesn't show any color difference. Means it doesn't stain with any either of base or acid. Okay, so in this what you have to remember, granulocytes, in granulocytes neutrophils are abundant. You can see its count is 60 to 65%, most abundant among the WBC cells. And neutrophils are phagocytic. Phagocytic means what? Whenever a foreign particle will come, no, at that time this neutrophil cells will engulf it, phagocyte, destroy it. Okay, so next one is eosinophils. Eosinophils, what is the function of eosinophils? It resists the function, it resists the infection. If any infection is occurring to the body, it stops it. Okay, associated with allergic reaction, if any allergic reaction is occurring, then it recognizes it and stop it. Okay, this is the function of eosinophils. Next, basophils. See, percentage of eosinophil is 2 to 3%. Okay, basophil are the least cells, 0.5 to 1%. These are the least WBC cells in number. Okay, what basophils will do? They will secrete histamine, heparin and serotonin. Histamine, heparin and serotonin, inflammatory reaction will occur. Okay, so next one is agranulocytes. Agranulocytes are lymphocyte and monocyte. Okay, lymphocyte see percentage 6 to 8%. Monocyte and neutrophils, these two helps in phagocyte, means phagocytic action. The same, if any foreign particle will come, these cells will engulf it and destroy the foreign particle and protect the body from foreign particle. Okay, so monocyte and neutrophils both are phagocytic, 6 to 8%. Next, lymphocyte. Lymphocyte, we all know that two type of cells are there, B cells and T cells. So what is the function of them? They provide immune response to the body. Okay, and its value is 20 to 25%. So quickly let us tell once again. WBC granulocytes are there, agranulocytes. In granulocytes, neutrophils 60 to 65% phagocytic. Eosinophils 2 to 3% resist infection and associated with allergic reaction. And basophil least in number 0.5 to 1 and release histamine, heparin and serotonin, inflammatory reaction. In agranulocyte, monocyte, that is phagocytic, lymphocyte, immune response. Okay, so this is in descending order in number. Okay, so that for easy remembrance, neutrophil 60 to 65, lymphocyte 20 to 25, monocyte 6 to 8%, eosinophil 2 to 3, basophil 0.5 to 1 least. Now let us discuss about platelets and or thrombocytes. Okay, see, in bone marrow, in the bone there is red bone marrow, no? In the bone marrow some megakaryocyte cells will be there. Okay, rupture of this megakaryocyte, some fragment of cells will be formed. Those are called as thrombocytes. Okay, what is the function of thrombocyte? Coagulation and clotting of blood. Okay, see, if platelets, clotting of blood is its function. If this platelet count is decreased, who will do the clotting? No, clotting disorders will occur. If the blood is not clotted, then what will happen? Excessive loss of blood will occur. See, this is from NCERT. RBC, we know red color RBC, and platelets, see, fragments type. And this is eosinophil. Eosinophil is a granulocyte, headphone shape, and neutrophil, that is multi-lobed. Okay, and basophil, basophil nucleus we can't see properly, it will be like this granulocyte type. Okay, and next one is monocyte. You can see single monocyte, a kidney shaped cell. Okay, kidney shaped cell. And these are lymphocyte, T lymphocyte and B lymphocyte. Okay, we are done with the composition of the blood, that includes plasma and formed elements. Okay, now let us move further into blood grouping. In blood grouping, there are two types of blood grouping, ABO blood grouping and RH blood grouping. First, let us discuss about ABO blood grouping. Mostly we will be doing both the blood grouping just to prevent the complications during blood transfusion. Okay, so first we have to remember how many types of blood groups are there. See, universally 4 types of blood groups are there, that is A blood group, B blood group, AB blood group and O blood group. And mine is O blood group, O positive. So you can comment your blood group, okay, in the comment section. So first we will discuss what is antigen, what is antibody and where they are present. Okay, first of all, antigens are there, no? Antigens are present on RBC. RBC we already discussed, red blood cells. Okay, now if it is A blood group, same antigen will be present on their cell, because now A antigens are present on RBC. That is why they are saying no, her blood group is A blood group. No, and if there is a B antigen on the RBC, then the blood group is B. And if A antigen is also there and B antigen is also there, then it is AB blood group. Okay, and if no antigens are there, one antigen is also not there, then what you have to remember, it is O blood group. Okay, that is why 0 O no antigens are present, okay, and antigens are present on RBC. Now antibodies, what are antibodies? Antibodies are proteins. These are proteins which are produced in response to antigens, okay. Now see, now our blood group is A blood group and antigens are A, and if so opposite antibody will be produced in the plasma. See, now A antigen A blood group, so anti B, B antigen B blood group, so anti A. Now A antigen is there, B antigen is there, all the antigens are there. What antibody will be there? If any of the antibody will be there, then agglutination will occur, okay. In the cell only agglutination will occur. Now antibodies and antigen reaction will occur. Just to not occur that, reverse antibodies, not the same antigen. See, antigen is A and antibody is B. If antibody is also A, then agglutination process will occur. That is why anti-B. B antigen means anti-A. Both the antigens are there, that is why no antibodies. And we know that 0, O blood group, no antigens are there. That is why both the antibodies are present, okay. One thing you have to remember here, that is, what is the donor blood group? Donor means O blood group is there, no. O blood group is given to any of the blood group people. Whenever they have emergency, no, O blood group is ready to give to anyone. See, it gives to itself and all the other blood groups, okay. Other thing what you have to remember is AB blood group is there, no. See, it is like, you can remember, it is larger in size, no. See, it is just individual digits, but it is larger in 2 digits are there, no. You can just alphabets are there, so you can just think that everybody is ready to give this AB blood group person. That is why it is becoming bigger, okay. So you can remember that A blood group, B blood group, O blood group and AB blood group, everybody is giving to this. That is why it is called universal recipient, and this is universal donor. And what are the blood groups giving? Who will be the donor blood group? A itself can give, and O is universal, you know, no. B itself, and O is universal. See, this is the remembrance way, like how you can remember. But actual the thing is, now if B donor will be giving its blood to A blood group, okay. So what will happen? B blood group person will be giving its blood to A blood group person. So here anti-B antibodies are there, okay, and B blood group person will be having B antigens. So this antigen and antibody reaction will occur, and this red blood cells will die, and it will cause complications, okay. You understand? In the same way here also. Now if we give B blood group, means same blood group can be given, and O is universal donor. But if A and A blood group person is giving the blood, then A antibody is there, and A blood group person has A antigen. So again there will be agglutination. Now in AB, everybody can give, no exception, because antibodies are only not there, so that any agglutination process will occur, okay. Now we know O blood group. See, both the antibodies are there. If you give A antigen A blood group, then this will cause agglutination. So that is why O gives to itself only, but O is like donor type, it gives to everyone, universal donor. And ABC, it is receiving from everybody. That is why it is becoming bigger. That is why everybody gives to AB, either B, A, O and AB itself. And A blood group people can give to A, and O is universal donor, B blood group can give to their B blood group people, and O is universal donor. Okay. So this is see the same thing. O blood group donate to person with any blood group. That is why O is universal donor, and AB group is there, no? It accepts blood from AB and also from other blood groups. That is why AB is universal recipient. Okay, we are done with ABO grouping, and next one is RH grouping. RH grouping is so easy. Now see, my blood group is O positive. Okay, O means in my RBC no antigens are present. So now see O 0, no antigens are there, and antibodies are anti-A and anti-B. Okay, that is why O. What is meant by positive? Positive, why it is there means in my RBC RH antigen is there, that is why positive. If my blood group was O negative, then no antigens are there, no A antigen, no B antigen as usually no, and RH antigen is also negative, that is why O negative. If it is positive, then RH antigen is present. Okay, now let me discuss here, you will understand better. So RH antigen is there, no? RH antigen is similar to the one present in rhesus monkey. In rhesus monkey also RH antigen is present. That is why RH grouping. That is why we named it as RH antigen. Okay. See, if on RBC surface RH antigen is present on the surface of RBC, then it is RH positive. And positive is there in 80% of humans. Around 100% of humans, 80% of people are RH positive. Okay, see, on RBC if RH is there positive, not there negative. Okay, and one more complication, it is famous complication. If you are studying RH grouping, then it is most important complication, that is erythroblastophetalis. What is erythroblastophetalis? When RH negative mother, she is RH negative means something AB negative, O negative, A negative or B negative, RH negative mother, and she married to RH positive father. Okay, that is why child will be RH positive. Okay, so RH negative mother and RH positive first baby, that will cause erythroblastophetalis. Okay, I will explain in detail how it causes. I'm just telling, remember, RH negative mother and RH positive child. See, RH negative patient exposed to RH positive blood, what happens? In RH negative patient, no antigens will be there and antibodies also will not be there. Okay, now RH positive blood is introduced in her. If RH positive means what? Antigens are present, no? So in the RH negative female, what will happen? Against that RH antigens, RH antibodies will be produced. Okay, that is what they are saying. RH negative patient exposed to RH positive blood, RH antibodies are formed. So that is why RH grouping is matched. This is also checked before blood transfusion. See, this is the main case, erythroblastophetalis. Okay, it is so simple, you will understand it very nicely, I hope so. So imagine imagine pregnant woman is there and she is RH negative. I told, no, mother is RH negative and this fetus is there, no, fetus, child is RH positive. Okay, at this time during pregnancy, generally what happens, when RH negative person exposed to RH positive person blood, then it has antigens, no? Against this antigens, antibodies will be formed. Okay, but between mother and the baby there is a placenta which separate the fetus from maternal blood. That is why there will be no exchange. Antibodies will not be formed. See, Rh antigen of the fetus don't get exposed to Rh negative blood of the mother in the first pregnancy because it is separated by placenta. But during delivery, what happens? See, during delivery, some leakage of the fetal blood will occur in the maternal blood. Okay. See, during delivery, exposure of the maternal blood into a small amount of fetal blood. Now what happens? Against Rh antigens of fetus, Rh antibodies will be produced by mother in subsequent frequency, means in the second pregnancy, third pregnancy. Okay. Now what happens? Now antibodies are produced, Rh antibodies. This baby came, okay, delivery occurred, and small amount of Rh antigen were there, no? Against that antigen, antibodies mother has formed. Now second baby also Rh positive. Then what happens? What happens means these antibodies are already formed. They will go and kill the Rh antigen RBCs. Agglutination process will occur. And baby will die. The complication will occur. That is called erythroblastofetalis. Okay, I think you understood. Destroy the fetal RBC and erythroblastofetalis will occur. That is fatal. Baby can die, or due to RBC damage, anemia may occur or jaundice may occur. Okay. See, that's what it is the first case. In the second case, Rh negative mom, but Rh antibodies are produced. How? During the last pregnancy, when the delivery occurred, some amount of blood, Rh antigen RBCs were there, no? That is why against that antibodies are produced. Now in the again subsequent pregnancy, Rh antigen, Rh blood group baby is formed. At that time, these Rh antibodies will cross. Okay, this antibodies will cross across the placenta and will cause agglutination and death. Okay. So what is the condition to avoid this erythroblastofetalis? What is the condition we have to apply? That is, immediately after first pregnancy, we have to give anti-Rh antibody. Okay, we have to give anti-Rh antibody. Let me explain what happens if we give anti-Rh antibody. See, now small amount of Rh antigen blood cells are there. Okay, RBCs are there. So before the mother make the Rh antibodies, what happen if we give this anti-Rh antibodies? So it will destroy these antigens which are left in the mother blood by the delivery of the first pregnancy. It will destroy these antigens. Before mother immune response will recognize this and produces this Rh antibody. Okay, I think you got this. So this is all about blood grouping, ABO blood grouping and Rh blood grouping. Let us move further to coagulation. Coagulation means what? Blood clotting. When there is injury in the body, excessive bleeding will occur. To prevent that excessive bleeding, coagulation will occur. Okay, see, blood exhibit coagulation or clotting in response to injury. Why? To prevent the excessive loss of blood. See, there will many factors will be released during coagulation. See, now injured tissue is injured, then certain factors will be released. See, calcium ion plays an important role in coagulation. See, injury occurs and that stimulate the platelets. Platelets are clotting factors, no? They release certain factors. Okay, in the presence of calcium, and mechanism of coagulation will occur. In the response to injured tissue and injury, platelets will release certain factors in the presence of calcium, and mechanism of coagulation occurs. See, so here main enzyme thrombokinase. Thrombokinase means, see, it is breaking down. Kinase means breaking. Prothrombin is there, no? Prothrombin in the presence of thrombokinase will become thrombin. It is inactivated form becomes activated form in the presence of calcium. Now this activated thrombin will act as an enzyme. It converts fibrinogen to fibrin. It is insoluble in the presence of calcium. And what happens? Fibrin is insoluble and it forms reddish brown clot which include fibrin and damaged platelets. Okay, this is the process. Thrombokinase is a certain factor and enzyme produced in response to tissue injury or injury, releases platelets, and mechanism of coagulation is started and thrombokinase is released. Prothrombin in the presence of calcium will form thrombin, and thrombin further act as an enzyme and converts fibrinogen into fibrin in the presence of calcium, and fibrin and damaged platelets will form reddish brown clot. One more thing which you have to remember is it is a cascade mechanism. Cascade mechanism means what? See, in the previous chemical reaction, one product is formed. This product is becoming an enzyme for another reaction, so it is helpful in another reaction. It is a cascade type. Okay, step by step everything is interrelated. Okay, and calcium plays an important role in clotting. Now let us discuss about lymph tissue also. See, lymph tissue means what? Now blood vessel is there, artery. Okay, it will break down into capillaries and gives oxygenated blood to tissues. So in between the tissues, see, some cells and water soluble substances will move out from the cell. From the cell they will move out in between the cells. Okay, and leave larger proteins and formed elements in the blood vessels. What is this fluid in between the blood vessels? In between the cells, what is this fluid which contains cells, some H2O and water soluble substances? That is interstitial fluid or tissue fluid. Okay, what is the composition? It is same as plasma because same mineral composition as plasma. Okay, so exchange of nutrients, gases between the blood and cells occurs through tissue fluid. Whatever there is exchange of gases or nutrients between the blood and tissue will occur through this fluid, that is interstitial fluid. Okay, now this interstitial fluid is there, no? It will be drained by lymphatic vessels. Okay, lymphatic vessels take this interstitial fluid and drain into a vein. This lymphatic vessels will be having lymph. Okay, what is lymph? Lymph is a colorless fluid. See, it is a colorless fluid and lymphocytes are present. What lymphocytes are responsible for? It is responsible for immune response. We discussed in WBC, right? A granulocyte, it is responsible for immune response. It is a colorless fluid. And lymph is also important carrier for transport of nutrients and hormones. This is all about lymph tissue. And you can see in interstitial villi, lacteal is present. Now fats are there, no? Fats are absorbed through lymph, that is in lacteal. Fats are insoluble and they can't be directly absorbed into the mucosa. That is why they are absorbed through lymph in the lacteal. So this is all about blood composition, RH grouping and ABO blood grouping and what is this coagulation, lymph tissue. Okay, now we are entering into circulatory pathway. This is so interesting, very, very interesting. Let us discuss. So, circulatory pathway, it is open and closed. What is meant by open? See, this is heart. Blood vessels are there. Blood will be transported by blood vessels, and it does not have minute capillaries. Okay, it will just drain into a sinus. Just in the body fluid it will be drained. That is why it is opened. Okay. Common example is arthropod and mollusk. What is meant by closed? See, this is general in humans also. This closed type of circulatory system is present. Heart, blood vessels, capillaries, tissues, then again venules, vein, and then heart. See, it is a closed type. Nothing is falling into a cavity or something like that. It is, what are the examples of closed circulatory pathway? Annelids and chordates. Okay. See, blood pumped through the large vessels. Blood pumped through the large vessels into open spaces or body cavity, that is sinuses. Okay. Blood pumped into closed network of blood vessels. See, it is a closed network of blood vessels. Okay. Here what you have to remember, all vertebrates possess muscular chambered heart. All the vertebrates are there, no, fishes, amphibians, reptiles, birds and mammals. All these vertebrates have muscular chambered heart for circulation. In fishes, only two chamber heart is present, that is one atrium and one ventricle. Okay, this is the heart of fish. In amphibians and reptiles except crocodile, three chambered heart, two atria will be there and one common ventricle will be there. Okay, and in crocodiles, birds and mammals, four chambered heart, two atria and two ventricle. Okay, now let us discuss the circulation pathway in the above vertebrates. Okay, in fishes what happens, heart is there, heart will pump deoxygenated blood. Gills is a respiratory organ, we discussed in respiration. Deoxygenated blood will become oxygenated blood by going near the gills, and oxygenated blood will supply to tissues and it becomes deoxygenated and goes back to heart. Okay, it is a single circulation in fishes. In amphibians and reptiles, what happens, two atrium is there, atria is there and one ventricle is there. All the deoxygenated blood from tissues will go into the right atria. This is right atria, this is left atrium. Okay, and oxygenated blood from lungs, skin and gills will go into the left atrium, and at last in the single ventricle both the deoxygenated and oxygenated blood will get mixed. And what is this, again it is supplied to tissues. Okay, this is incomplete double circulation, it's not double circulation. See, at the end mixed blood is supplied. Now in birds, mammals and crocodile, double circulation is there. How? Deoxygenated blood from the tissues goes into the right atrium and right ventricle, and the deoxygenated blood will go to lung for purification. From the lung, purified O2 will come into the left atrium, left ventricle, and the oxygenated blood will go to tissues. Two separate circulatory pathways without mixing of blood, double circulation. Okay, now let us discuss about human circulatory system. Okay, human circulatory system include heart, blood vessels and blood. Blood is a fluid, we discussed its composition, everything. Blood vessels are network of closed and branching. It is closed and branching type blood vessels. Okay, heart is a muscular chamber, it is muscular chamber, muscular chambered. Okay, first let us discuss about heart. Heart is mesodermally derived. See, it is organ. Okay, it is a mesodermally derived, situated in thoracic cavity. See, in the thorax lungs are there, no, in between the lung heart is present, and it is tilted towards left. This is right lung, left lung. In between the thoracic cavity, between the lungs, it is tilted slightly to the left lung, left side. Its space is called mediastinum. Okay, situated in the thoracic cavity between the lungs, slightly tilted to left. See what is the size of the heart? Clenched fist. If you close your fingers and thumb, the size you can notice, that is called clenched fist. Okay, this heart is covered by double membranous bag. Okay, that is called pericardium. Okay, in between the pericardium, pericardial fluid is there. Okay, so this is all about human circulatory system. Means we are discussing about the heart. Okay, now let us discuss. This is so interesting, students. If you listen to me carefully, I think this will become a favorite topic for you. So heart has 4 chambers in humans, we discussed earlier. 2 atria and 2 ventricle. Atria, see, these are atria, right atrium, left atrium. These are smaller upper chambers, and ventricles are right ventricle, left ventricle. These are larger lower chambers, lower side present. So here let me say some septas are there. Okay, see, these are the chambers, they can't be freely present. Some strong septas should be there, connective tissue should be there to adhere them. Okay, see, in between both the atrias, I will just write right atrium, left atrium, right ventricle, left ventricle. Okay, so okay, between the right atrium and left atrium, some thin muscular wall is there. Thin muscular wall, that is called interatrial septum. It is between, intermediate we say, no? Between higher education and schooling, intermediate will say, no? In the same way, it is between 2 atria. That is why interatrial septum. See, between 2 ventricles, there is a thick wall. Between atria, there are small atrium, no? That is why thin wall is enough. But these are very larger ventricles. That is why thick wall will be there. This is called interventricular septum. Between the 2 ventricles, that is why interventricular septum. Okay, and not only this, in between the atria and ventricle also some muscular wall is there. This is also very thick, should adhere nicely. It is a thick fibrous tissue. Okay, it is a muscular wall. It is a fibrous tissue in between atria and ventricle. That is atrioventricular septum. Atrioventricular septum. Okay, so between, see this, you understood, no? Interatrial septum, interventricular septum, and atrioventricular septum. This atrioventricular septum will be having some opening. Okay, now blood should flow, no? Blood from vein will enter into right atrium and then into right ventricle, no? There should be flow of passage. It is thick fibrous, we know, but there is some opening. Okay, through that opening, 3 muscular cusp or flaps will be there, guarded by 3 muscular cusp or flaps, that is called tricuspid valve. Okay, in the left atrium and left ventricle, same opening will be there, and it is guarded by 2 cusp or flap. That is why 2 muscular flaps or cusp, that is mitral wall or bicuspid wall. It is tricuspid wall, it is bicuspid wall, because 2 cusp, 3 cusp, bicuspid and mitral wall. Okay, so here now from ventricles, from right ventricle, pulmonary vein, it supplied deoxygenated to pulmonary vein, no? Here also it is guarded by 1 valve, that is called semilunar valve. Now left ventricle is there, no? It supplies oxygenated blood to aorta. Okay, in between aorta and left ventricle also some valve is there, that is called semilunar valve. These both are semilunar valve. This is tricuspid, this is bicuspid or mitral valve. Okay. So this is what, see, atrioventricular septum. Septa of each side has a opening through which atrium and ventricle are connected. This is what it is. Valve in heart allow the flow of blood in one direction. This valve is there, no? This will allow flow of blood in one direction, no back flow. It prevents the backward flow. Okay, one direction process should be there. That is why valves are present. Now if blood is present in right ventricle, it can go to right atrium, no, but to prevent that, valves are present. See, atria to ventricle and then pulmonary artery. See, atria to ventricle, then aorta. Okay, this is the structure and its function. Cardiac muscle means what? The muscle which is self-excitable and self-contraction it can do. Okay, now see, this wall of ventricles are there, no? These are little bit thicker. These are thicker than atrial walls. Why? Now blood will flow into atrium and from atrium the blood will flow into the ventricle and it should supply, no, this should supply the blood to the lung and this should supply blood to the what? Tissues, right? So this require more energy to pump the blood. Ventricles require more energy to pump the blood. That is why walls of ventricles are thicker than atria. Okay, see, in the heart it is made up of cardiac muscle, but there is a specialized cardiac musculature. One cardiac musculatures, special cardiac musculatures are distributed in the heart. That is called as nodal tissue. Okay, nodal tissue means this is a specialized cardiac muscle, auto-excitable. Okay, auto-excitable cardiac musculature, specialized, that is called as nodal tissue. So what are those structures, specialized cardiac musculature structures in the heart? Those are SAN. SAN means sinoatrial node, AV node and AV bundle. See, this is sinoatrial node which is located on the right upper corner. See, this is right side, this is left side. On the upper corner, upper side, right side, you can see sinoatrial node, and this is lower side of the atrium, and it is left side, left corner of the atrium, left lower corner. See, lower side. This is sinoatrial node is on the upper corner and AV node is in the lower corner, which is towards the left of the atrium. And after that you can see this sky blue, you can see this cyan color. These are called AV bundle. From AV node, AV bundle will be released. Okay, these are bundle of nodal fibers. Okay, AV node. See, from AV node, they are saying the distribution of the AV bundle. Okay, see, from AV node, this is what? Atrioventricular septa, no? From AV node, it crosses the atrioventricular septa, and in between both the ventricles, interventricular septa is there. On the top of the interventricular septa, this is present. AV node, atrioventricular septa, and on top of interventricular septum, what is present? AV bundle. And on the top of the interventricular septa, right and left bundles are formed. Okay, this right and left bundles will give rise to Purkinje fibers. What are the specialized cardiac musculature? That is sinoatrial node, AV bundle, right and left bundle branch, and Purkinje fibers. See, This nodal musculature is auto-excitable. It can stimulate by itself. Okay, it can contract the heart by itself. No external stimuli is required. Okay. And how it contracts the heart? It produces action potential. It produces action potential. Through that action potential, contraction of the heart will take place. And number of action potential vary at different part of nodal system. Now here little amount of action potential will be produced. Here another number, okay, different numbers, not same number of action potential will be distributed in every part of the nodal system. Okay, different action potential, different number will be produced, but maximum number of action potential is produced by sinoatrial node, that is 70 to 75 per minute. Okay, this sinoatrial node is there, no, that will help in initiating and maintaining rhythmic contraction of heart. Now sinoatrial node will initiate the contraction, okay, and also it will maintain the contraction also. That is where rhythmic contractions will occur in the heart. That is why it is called as pacemaker. The number of action potentials produced by sinoatrial node will be same as the heartbeat. See, 70 to 75 per minute. So this is all about specialized cardiac musculature, that is nodal tissue distributed in the heart. Okay. Now we will discuss cardiac cycle. Actually this is so conceptual, and if you understand, this is so easy. So you have to remember cardiac cycle is about 0.8 seconds. Okay, there are 3 stages. First stage is joint diastole. Joint diastole means, in the name itself, diastole means what? Relaxation. Joint means both the atria and ventricles are relaxed. Okay, this carries 0.4 second, and after both the ventricles are, both the chambers are relaxed, atrial systole will occur. This is about 0.1 second. Okay, after atrial systole we see ventricular systole. After atrium, atria will relax and ventricle will start contracting. That is about 0.3 second. So total 0.3, 0.1 and 0.4, that is 0.8 second. So let us see what happens in joint diastole. In joint diastole, all chambers will relax, I told you. And now what happened? This AV valves will be there, no? Like atrioventricular valves, they will be opened. All chambers are relaxed and valves will be opened. Now what happens? Now valves are open, no? From vena cava, this is right side, this is left side, okay, of the heart. Now from vena cava, blood will flow into the right atrium, and as this tricuspid valve is open, the blood will flow into ventricle. Small amount of blood, okay, because it is joint diastole condition. But here you can see semilunar valve is closed, so blood will not go further into the pulmonary artery or aorta. Okay, here you can see pulmonary vein from the lung will enter into the, see, pulmonary vein, blood will be entered into the left atrium, and as bicuspid valve is open, it will enter into the left ventricle. Okay, but it can't go further because semilunar valve is closed. Okay, see, semilunar valve is closed. This is the condition of joint diastole. Now what happens? All the chambers are relaxed and flow of blood will occur into ventricle, till ventricle. Now SA node will generate action potential. SA node is present on the right upper corner of the atrium. Now, if action potential is generated, contraction will occur. Whose contraction? Atria, because it is present on the right upper corner of the atrium. Okay. After joint diastole condition, SA node will generate action potential. Now see, in atrial systole, which is about 0.1 second, this atria will contract. Now blood inside it will be there. No, it will be pouring into ventricle 30 times more. See, both the atria contract simultaneously and increase in blood flow to ventricles by 30%. In both the ventricles, how much the blood flow will increase? 30%. Okay, this is all about atrial systole. Now what happens? Now the SA node generated the potential and atria contracted and 30% blood flow into the ventricles will occur. Now this AV bundle is there, no? Lower left corner of the atrium. AV node will generate action potential. AV bundle will generate action potential. AV bundle and branching bundles will generate, and Purkinje fibers will generate this action potential to ventricular side. Okay, action potential is generated, conducted to the ventricular side by AV node, AV bundle and Purkinje fibers. Let us see what happened in ventricular systole. This is also conceptual, and the cycle occurs for 0.3 second. See, now action potential is generated, so both the ventricles will contract. First they will contract, name itself systole, means contraction. Both the ventricles will contract simultaneously. When they will contract, some pressure, of course no volume will be decreasing because contraction occurs, and pressure will increase. Ventricular pressure will increase. Now pressure is increasing. See, now ventricles are contracting, so pressure will increase. Pressure will increase means the blood which will be present in this ventricles, no, it will try to go backside into the atrium. That is why what will happen, these valves are there, no, tricuspid and bicuspid valve, they will close, so that what happens, this blood should not go into the atrium. Now what happens, ventricular contraction will be even more. That's why here valves will be there, no, semilunar. This semilunar valves will open because they can't tolerate the pressure inside the ventricles. That is why it will flow through the pulmonary artery, and this blood will flow into the aorta. Now what happens, blood has flowed into their capillaries. Okay, now what happens, now pressure will decrease. Now in the ventricles pressure start decreasing, and pressure start decreasing, so blood has flown into this, and after that semilunar valve is there, no, that also will start closing because the blood is flowing into this capillaries, that is aorta, and to prevent backflow, see, semilunar valve will close. See, semilunar valve will close to prevent the backflow. Okay, now what they are saying is further pressure will decrease. Okay, so what will happen, now pressure is further decreasing, automatically this valves will be opened. Okay, atrioventricular valves will be opened, and what will happen, relaxation will happen. I will explain. See, now I think you little bit understood what I meant to say. Now once again I will explain. Now AV node will generate action potential and these ventricles will contract. Okay, now what happens? Pressure increases in the ventricle. If pressure is increasing here, to prevent the backflow of the blood into atrium, these valves will close. Now further pressure is increasing, so to release the or decrease the pressure, this semilunar valves will open so that the blood will flow into the pulmonary artery and aorta. Okay, now what happens? Now see, semilunar valves will open and flow of blood to the pulmonary aorta. Now what happened? Atria was contracting in the previous stage, no? Now ventricular systole is there, no? Ventricular contraction and atrial relaxation will coincide with each other. This is very important. Now what happens? The blood has flown into the pulmonary artery and aorta. So now ventricles start relaxing. Now they are relaxing. Then what happens? Pressure decreases. That's why what happens? Semilunar valve, I told, no, they will close because to prevent the backflow. So first this will close and further pressure decreases. That's why atrioventricular valve will open because it's relaxation state. This atria is also relaxed. Pressure is decreased. That's why generally AV valve will be opened only. In diastole you saw AV valve will be opened and semilunar valve will be closed. Okay. And again what happens? Again joint diastole condition will occur. See, I will show you once again. In ventricular systole, what happened? Atrial systole was occurring. Both were contracting and increases the blood flow by 30%. Now both the ventricles contract simultaneously and pressure increases. Ventricular pressure increases and AV valve will be closed to prevent the backflow. During this closure, one sound will produce, that is lub sound. Okay, through stethoscope we can hear this sound. What happened? Now this is closed and pressure in the ventricle still will increase. That's why semilunar valve will open and blood flow to the pulmonary artery and aorta will occur. Now what is the important statement? Atria diastole and ventricular systole coincide. In this ventricular systole stage only atrial diastole also will occur and it coincide. Now what happened? Contraction of the ventricle is over because blood flow has occurred. Now ventricles will relax. Okay, and pressure decreases. As pressure decreases, pulmonary semilunar valves are there, no, that will close to prevent the backflow. It will close. That is called as dub sound. Okay, this is also heard by stethoscope. Lub sound is closure of AV valve and dub sound is closure of semilunar valve. Okay, to prevent the backflow. Now further pressure will decrease, ventricular pressure, and AV valve as usual opens because everything will be in joint diastolic condition. So again there will be a repeat of the cycle. Okay, see here it is in short. 0.4 second is joint diastole, in which all the chambers are in relaxed state. AV valve is open and from vessel it supplies to atrium and through the ventricle. Okay, semilunar valve is closed in joint diastole. In atrial systole, atrial contraction will occur. How? SA node auto excitation will occur. Okay, and 30% blood flow increases to the ventricle in atrial systole, and ventricular systole is 0.3 second. And what happens? In the beginning only AV valve closes, that is lub sound production, and semilunar valve closes, tub sound production. In ventricular systole, both the ventricles will contract simultaneously and ventricular pressure will increase, through which what happens, AV valves will close to prevent the backflow, and further pressure will increase and blood will go into the pulmonary artery and aorta. Okay, now what happens, atrial diastole and ventricular systole will coincide. Okay, now the blood has flown into the pulmonary artery and aorta. Now what happens, this ventricles will relax because its function is completed. When ventricles are relaxed, pressure will decrease. Okay, and semilunar valve will be closed to prevent the backflow. Okay, at that time double sound we can hear. Okay, and further pressure decreases, then this AV valve will open and everything will be in diastolic condition. Atrial relaxation, ventricular relaxation, and AV valves are open and semilunar valve is closed. Okay, I think this is crystal clear now. See, one cardiac cycle 0.8 second. I told you heartbeat coincide with the action potential of SA node, that is 72 to 72. So number of cardiac cycles are 72 cardiac cycles. See, 70 to 75 per minute, but on average 72. What is stroke volume? Each ventricle is there, no? Each ventricle will pump out the blood during one cycle. Okay, that is called stroke volume. Each ventricle, remember, that is 70 ml. What is cardiac output? Stroke volume into heartbeat is cardiac output. Okay, remember one cardiac cycle is 0.8 second, 72 cardiac cycle per minute. Okay, stroke volume means each ventricle pump out the blood 70 ml. Cardiac output is stroke volume into heartbeat. Stroke volume is 70 and heartbeat is 72. Total you will get 5040 ml, approximately 5 liter. Okay, now there are two pathways, no, autonomic pathways. Neural system, autonomous system will be there, autonomic nervous system. In that you will find sympathetic and parasympathetic. Sympathetic system, what happens, every time sympathetic means it will show sympathy and the value will increase. Okay, see, cardiac output will increase, stroke volume will increase, heartbeat also will increase. See, cardiac output, stroke volume, heartbeat. In this formula only these three things are there. In sympathetic these three will increase. In parasympathetic, cardiac output decrease, stroke volume decrease and heartbeat decreases. Okay, this is all about cardiac cycle. Now electrocardiogram. See, never confuse between electrocardiogram and cardiograph. Graph means what we will be thinking, graph means that is a paper and graphical presentation, but no, in cardiac graph means a machine and gram means a graphical presentation of electrical activity of heart during cardiac cycle. During cardiac cycle, what is the electrical activity of the heart, that is shown in graphical presentation, is called electrocardiogram. See how this ECG is produced. Patient is there, no, we have to connect to machine by 3 electrical leads. Okay, 3 electrical leads should be connected to patient to the machine, that is one on each wrist. On each wrist one and one to the left side of the ankle. Okay, that there we can monitor the heart activity. Now detailed evaluation, if you want, multiple leads should be placed on the chest region, then you can see detailed evaluation. But generally what, 3 electrical leads, one each on wrist and one on left angle. Okay, see, this is the diagrammatic presentation of ECG. It includes 3C: P wave, QRS complex and T wave. In this only I have explained everything. See, P wave represent, first what happens, atrial systole will happen. Same here, P wave means atrial systole. And QRS complex. QRS complex means what? Ventricular systole will occur. Okay. See, ventricular systole will not start from Q. Okay. After Q wave, see, like this slowly it will start and continues till the end of the T wave. See, ventricular systole, at the end of the T wave, ventricle contraction will be occurring after, little bit after Q wave. Okay, what is T wave? T wave is ventricular diastole. Okay, atrial diastole in between the ventricular systole only will occur. It will be hidden. Okay, now see, let us discuss. P wave means, what did I tell you? Atrial systole. See, contraction of the atria, that is atrial systole. What happens here? Depolarization of atria. Depolarization of atria means electrical excitation of atria will occur and atrial contraction will occur. See, atrial contraction, atrial systole, electrical excitation. Now QRS complex, that is depolarization of ventricle. Depolarization of ventricle means ventricular systole, ventricular contraction. See, that starts slowly after Q wave. After Q wave it will start. This is Q wave, no? From here it will start. Start slowly after Q wave and marks the beginning of systole. This will be the beginning of systole, and end of T wave mark the end of the systole. This is end of systole. This is ventricular systole. Okay. See, and T wave is, this is depolarization because contraction is occurring. But ventricular diastole means repolarization. Repolarization of ventricle. Ventricles are excited to normal. They come back to normal. That is, relaxation of ventricles will occur. Okay. End of T wave marks the end of systole. See, end of T wave is end of ventricular systole, and T wave is repolarization of ventricle means relaxation of ventricle back to normal from excited state. Okay. Here you can see, by counting the number of QRS complexes in a given period, determine the heartbeat of an individual. In ECG, many see again QRS complexes will be there. P wave, QRS complex, T wave, then P wave, QRS complex. Like this, if we calculate the QRS complexes in the electrical activity of the heart, in a given period, if we calculate the number of QRS complexes, we can determine the heartbeat of an individual, because each of this one is PQRS T. This is, it represents one cardiac cycle. In that way we can calculate number of heartbeat. ECG obtained from different individual is roughly same. Okay, if you find any deviation, then there is abnormality or disease. That is why it is great clinical significance. Now let us go into double circulation. This is the third last topic we are about to end. See, in double circulation, first let us learn about the blood vessels. Okay, blood vessels has 3 layers. We know tunica intima means inner layer, tunica media middle layer, tunica externa external layer. Intima means inner lining of blood vessels by squamous epithelium. Remember, inner lining is squamous epithelium and middle layer is by smooth muscle. Contraction should be done, no? not contractions I can say, but media is of smooth muscle and elastic fibers, and externa is external layer of fibrous connective tissue and collagen fiber. See, internal layer squamous epithelium, middle layer smooth muscle and elastic fiber, external layer fibrous connective tissue and collagen fiber. Here one point you have to remember is tunica media is there, no. See, this is tunica media. In vein is small, and in artery see how much thick it is. Tunica media is thin in vein and thick in artery. And you all know this is the water double circulation. We discussed there, from the organs, deoxygenated blood through superior and inferior vena cava will go into the atrium and then into the ventricle through pulmonary vein. It will take the deoxygenated blood to lungs. From lungs, it will get purified. Okay, deoxygenated blood will get purified through pulmonary artery. It will come back into the atrium. See, from ventricles it's going to lung through pulmonary vein, and away from lung, oxygenated blood through pulmonary artery into atrium. This is called pulmonary circulation, going and coming back to heart. Okay, now this oxygenated blood will go into the ventricle, to the aorta, and then to the organs. From organs, deoxygenated blood to superior and inferior vena cava into the atrium. This is called as systemic circulation. System, different parts of organs, it is supplying blood, supplying blood and taking blood to the away from the heart. So it is systemic circulation. And one more thing which you have to remember here is, now intestine will be there, no. Intestine contains deoxygenated blood. It won't directly enter into the venules, vein and superior inferior vena cava. Okay, the blood will go through a duct to the liver, and again all the deoxygenated blood of intestine and liver both will be carried through the inferior vena cava. That is called as, that vein is called as hepatic portal vein. It is going near the hepatic, means liver, that is a hepatic portal vein. Okay, directly can't drain into the vein, that's why it will drain its deoxygenated blood into the liver. See, special coronary system is present in the heart, which arteries will originate from the heart and supply to the heart. Special system, circulation of blood to and from cardiac musculature. This is called as double circulation. Now let us learn about the regulation of cardiac activity. See, cardiac activity is intrinsic only. It generates its own action potential by nodal tissue, auto-regulated intrinsically. That is, myogenic heart can contract by itself. But some influence will be there. Neural center will be there, no? In the medulla, you know that this is a forebrain, midbrain, and this is pons and medulla cerebellum. These are hindbrain structures. In the medulla, you can find one neural center. Okay, neural center in the medulla. Through autonomic nervous system, I told you autonomic nervous system is divided into sympathetic and parasympathetic, no? Through sympathetic system, heartbeat will increase, cardiac output will increase, and strength of ventricle contraction also will increase. Whereas in parasympathetic, heartbeat decreases, cardiac output decreases, and speed of conduction of action potential will be decreased. Action potentials are, speed is decreased. That's why what happens, contraction strength also will decrease. Okay. Some adrenal medullary hormones will be present which will increase the cardiac output. So this is the last topic, that is disorders of the circulatory system. First one is high blood pressure. Normal blood pressure is 120 by 80. 120 is systolic blood pressure and 80 is diastolic or resting pressure. Okay, systolic or pumping. 120 mm of Hg. Okay, remember it is mm of Hg. 80 mm of Hg. What is high blood pressure then? When blood pressure is more than 140 by 90. These are the values it should be having, or more than 140 by 90, it is called hypertension. Means heart disease will occur and also affect the kidney and brain. Okay. Now high blood pressure is one disorder. Next, coronary artery disease, that is CAD. What is meant by this? It is also called as atherosclerosis. Now blood vessels are there, no, which supply to the heart. Blood vessels are there. Now what happens? Deposition of calcium, fat, cholesterol, fibrous tissue, they will form a layer in the blood vessels, and it will become narrow, see, and it will affect the blood supply to the heart, and inadequate blood supply will occur in the heart. That is called as coronary artery disease or atherosclerosis. Now blood supply is decreased, no. Just imagine this as sequence. First chest pain will occur, okay. Angina pectoris means what? Acute chest pain. Why chest pain is occurring? Oxygen reaching the heart muscle is decreasing, okay. That is why no or decrease in oxygen reaching to the heart muscle. That is why angina will occur, angina pectoris. It will occur in men and women of any age, but commonly elderly people and middle-aged people. Okay. Condition occurs that affect the blood flow. Blood flow complication is occurring. That is why O2 is not reaching to the heart muscle and acute chest pain is occurring. Okay. These are the common terms where you get confused, but today I will make it clear. Angina, we told, oxygen supply is lesser, and that is why chest pain will occur. Coronary artery disease is due to narrowing of the vessel, due to deposition of these components, blood supply is decreased to the heart, no. Heart attack. Attack means what? Heart muscle is damaged by inadequate blood supply. After angina, heart attack will occur. These are the steps. Due to inadequate blood supply, what happens? Heart muscle is damaged and heart attack will occur after angina. Heart failure means what? First of all, what is the function of the heart? Pumping of the blood. Now failure, it is failing its activity. Okay, that is why heart failure means heart is not pumping the blood effectively to meet the need of the body. Sometimes it is called as congestive heart failure. Why? Congestion of lung is the main symptom. That is why congestive heart failure. Okay. What is cardiac arrest? Think that somebody is arresting the heart. That is why it can't beat. It is completely arrested, closed and compact. That is why it is not beating. Heart stop beating. Okay. And I think now it is little bit with this understanding you might remember. See, I will say once again. Heart attack means what? After angina, due to inadequate blood supply. See, inadequate blood supply, heart muscle will damage. Now heart failure is what? Failure, it does not do its function, not effectively pumping the blood. Cardiac arrest means what? Somebody is arresting it. Okay, no space, full congested. So heartbeat stops. Okay. Thank you. From here, it's the end of the blood and circulatory system. I think all the concepts I made it clear and crystal clear. So we are done with the body fluids and circulation. I hope I reached to you and you understood all the concepts. After reading this unit from this video, please try to read the NCERT. Now I think NCERT you can complete in 15 to 20 minutes maximum with all the concepts clear. Okay, thank you.